what we're realizing now that most of cancer is really a kind of a genetic family tree. We used to think that how cancers came was one cell would get a mutation and then get another mutation, and then you became a cancerous clone, and that whole clone was fairly homogeneous. We now know that, in fact, if you look at in most types of tumors, there's a so-called intratumoral heterogeneity, where there's actually multiple clones at present at diagnosis, and they're related to each other, but they aren't genetically identical to each other, and their behavior can be different. So that some clones can be sensitive to chemotherapy, and some clones can be resistant. And we think that the natural history of most cancers, which are to respond and then relapse, is really this Darwinian selection taking place, where the first clones there that are sensitive, they get wiped out, and that just creates more room and more food to eat for the resistance clones. So CML is always thought to be a fairly homogeneous disease because every patient has the Philadelphia chromosome, the juxtaposition between the chromosome 9 and 22, and in mouse models that seems to kind of drive the biology. But now we know by more sensitive studies and the like that there actually is a fair amount of heterogeneity even at diagnosis, so that patients with a Philadelphia chromosome can have other mutations, particularly ones that you see in other myeloid malignancies. And moreover, these may associate those patients who have a worse response, either more aggressive disease and go to accelerated phase and blast crisis, or who just have a poor treatment response in chronic phase. And so now we're seeing that, in fact, the types of heterogeneity that we are seeing in all these other cancers, we're starting to see some of that in CML, and we're trying to now understand how we can parlay that into picking out patients who might need a stronger agent, ones who might need to change TKIs more rapidly, patients who we may have to add a second drug on to get a great treatment response or not. And these methods that we're using now to really track this type of information is both by more sensitive sequencing techniques. Most next-generation sequencing techniques can pick out one mutation in about 100 normal. There are ways, so-called duplex sequencing is one of them, where you can get down to a resolution of finding a mutation in 1 million cells. So that's going to allow us to kind of get an idea of the complexity that is there at baseline under the radar for how we look at these things now. The other thing that's coming to the fore now is so-called single-cell sequencing. It's obviously hard to understand the genetic complexity of a disease when you grind it all up together, because you're just kind of getting the average of all the clones as far as mutation load and the like. There's methods now where you can actually separate cells into single cells and actually do the genotyping on each cell, so you get a much better understanding of really the complexity that underlies the biology. And now the next thing is going to be is how do we drive that into treatment management? Because we'll be able to soon follow not only the disease burden, but when you have residual disease, you can be able to tell not only how much residual disease is there, but what types of residual disease, what types of mutations are selected for, so you can potentially target those mutations to get a better response.